Good afternoon, colleagues. Good morning, our friends from the US. And of course, good afternoon, good early afternoon for the colleagues from Oxford. Allow me to take this early opportunity to welcome you to this seminar, which is a little biased to handle cancer. And of course, we had a running title, which is the hallmark of cancer towards identifying research gaps. Our guest speaker is a friend of this university, Dr. Victor Oria. Victor holds a PhD in cancer biology from the University of the Freiburg, Germany. And he also holds an MSc in medical diagnostics from Cranfield University, UK, and a BSc in biochemistry from this very university, Kenyatta University. He has a considerable research experience in the field of cancer biology. Victor's main research interest at the moment is unmasking the genetic and molecular events within the tumor microenvironment that drive the cancer metastasis. Currently, Victor is also a postdoctoral fellow in the lab of Dr. Lucia Evasion at Yale's Cancer Center in Yale University in the United States. Again, now he's actively focusing on the microenvironmental factors that promote escapes from tumor dormancy in metastatic melanoma and subsequent colonization of the brain. Victor also serves as the acting chief scientist and head of research division at the Cancer Research Kenya, which is currently registered as the Integrated Cancer Research Foundation of Kenya, where he is responsible for directing the organization's scientific strategy. It's also good to note that Cancer Research Kenya is a young organization that last year with a sole mandate in combating cancer through evidence-based basic and translational molecular oncology research. This is clearly a very a niche in Kenya that is not really overly researched. So without further ado, this seminar has registration of approaching 130. At the moment, I see we have around 41 already have joined us. It will run for an hour. Victor will give us a bit of intro and a lot more on the real stuff. And of course, for 30-40 minutes, precisely 40 minutes really, and we will have 20 minutes for back and forth, the question and answers. So this, for us, we look at this as a platform for expanding knowledge and, of course, identifying critical areas that we can plug on as researchers to see whether we can develop largely the collaborations through the external networks, and also what can also be of mutual benefit for us as researchers in this university. During the presentations, we'll still just listen and you note your questions, and at the end of it all, we'll have a back and forth for some time. So we have different institutions here at Oxford. We have Yale University, we've got local universities, we've got a number from Kenyatta University, and we are privileged to have this happening. Over time, we will be having specialized conversations like this, and I would urge colleagues and to thank you in advance for joining, and let's keep this discussion going forward. Victor, when you're ready.
Okay, I'll try to share my screen and then tell me if you can see it. Okay, the entire screen. Can you see my screen?
Yes, we can.
Do you see the presentation?
Yes, but not yet in presenter mode, just your slides.
Okay, all right. So thank you very much, Richard, for the kind introduction. So yeah, today I'm going to be talking about the hallmarks of cancer towards the identification of research gaps, specifically in Kenya. And as Richard has said, I work in the field of melanoma brain metastasis. However, I'm not going to be presenting anything on that. Rather, I'm going to be giving a general overview of the hallmarks of cancers, because I think this is very important for the understanding of any future niche-specific topic that we might be presenting here to the very same audience. So my presentation is divided into three parts. The first part will be a brief definition of the hallmarks, and then the second part I'll look at some of the hallmarks in a bit of detail and try to explain what they mean. And then in the last section, we look at the research gaps in Kenya and how we should address them. So when we speak about cancer, is it a homogeneous disease? Yes, to many people not experts in the field, we might think of cancer as a homogeneous disease, but research tells us otherwise. Cancer is very complex and diverse in terms of genetics, in terms of pathology, histology, how it responds to therapy, and even the prognosis. However, underlying this complexity and diversity are common pillars which are referred to as the hallmarks of cancer, and this will be the subject of the presentation today. So in figure A, you see the simplistic view of cancer, what most of us might think cancer is, where you just have a bunch of cells growing. But in the real sense, cancer, the real situation for cancer is under figure B, where you have this whole kingdom with different cell types that try to support the growth and progression of this disease. So among the cell types that make up tumors, you have the cancer cells, which are epithelial in nature. Then you have the cancer stem cells, which are responsible for tumor regeneration. Then you have cancer-associated fibroblasts or CAFs, which form part of the tumor stroma or the microenvironment surrounding the tumor. And then we have the endothelial cells and pericytes that form part of the blood vessels that supply the tumor with oxygen and nutrients. And then we have immune cells, and immune cells may play a dual role. Some may support the growth of the tumor, and some might actually try to stop the tumor from growing. So going forward, I want you to have figure B in your mind as the actual view of all tumors that affect humans. So in the year 2000, Douglas Hanahan and Robert Weinberg described the 6 hallmarks that were common to all tumors that were under research. And 6 of these hallmarks include sustaining proliferative signaling, evasion of growth suppressors, resistance to cell death, induction of angiogenesis, enabling of replicative immortality, and activation of invasion and metastasis. 11 years later, they introduced 2 new hallmarks, that is deregulation of cellular energetics and evasion of immune destruction. So these hallmarks are very important because they are the driving engine towards tumor growth. And it's very important for me to say at this step that not all tumors rely on these hallmarks. Each tumor might rely on 1, 2, or 3 specific hallmarks depending on the nature of the tumor. So for the next few slides, I'll be talking about 5 of these hallmarks. So I'll just start with the first one, sustaining proliferative signaling. So our cells usually need signals from the microenvironment that instruct them to grow and divide. So cancer cells have identified some of these signals and have hijacked normal genes that are part of these pathways that then stimulate uncontrolled growth cycles that drive cellular proliferation. And these normal genes undergo what we call activating mutations ensuring that they are always turned on. And two of these genes that are specifically mutated include the RAS GTPase and the RAF kinase, and you'll see them in the next slide in a minute, which are mutated in different forms of cancer. So, for instance, the RAS GTPases are mutated in pancreatic cancer, in lung cancer, in bladder cancer, and melanoma, while the RAF kinases are seen to be mutated in melanoma, liver, and kidney cancers. So what are the functional implications of these mutations? So on the left side, you see normal cellular signaling. You have a growth factor binding to its receptor on the cell membrane, which then activates RAS GTP, that then activates BRAF, which then phosphorylate MEK and ARC, leading to normal cell proliferation and survival. So this pathway has got a checkpoint. The checkpoint is that if there is no growth factor binding to the receptor, then the pathway is off and the cells don't grow or divide. So what do tumor cells do? They do what is happening here on the right side. They have a mutated RAF, which is in this case BRAF, which is activated. So it means the pathway is always on. So this activated mutation mimics the presence of a growth factor on the receptor, ensuring that the cell growth and proliferation is always on. So this is what happens in the context of melanoma. In the context of, say, pancreatic cancer, instead of RAF being mutated, you have RAS GTP that is mutated. And so this always switches the pathway on, even in the absence of a growth factor binding to the receptor. So for BRAF mutations, we have several approved inhibitors that are, for instance, sorafenib, which is used for lung cancer and kidney cancers, and vemurafenib, which is used to treat late-stage melanoma. However, for RAS mutation, we don't have any approved inhibitors yet. And this explains why a disease such as pancreatic cancer is very difficult to treat, because it's hugely dependent on Keras mutation. And therefore, the only way that we try to treat pancreatic cancer is either through surgery, radiotherapy, or chemotherapy, because it can't respond to any of the current inhibitors present in the clinic. So the second hallmark that we look at is evasion of growth suppressors. So, like I said in the first hallmark, that cell growth is a highly regulated process. So it means that there are checkpoints at every stage to ensure that cell cycle, cell division goes without any problem. So our cells have different prominent growth suppressors. Our cells have different growth suppressors, including P53, which is found mutated in 40% of all cancers. We have retinoblastoma protein, which is very important in the cell cycle process. And we also have APC and VHL, which are growth suppressors that are mutated or lost in either colorectal or renal cancers, respectively. So let's look at P53, for instance, and just try to explain what it does. So when a cell is stressed, or under normal physiological conditions, P53 is an inactive state. It's bound to this protein here called MDM2 and subject to proteasomal degradation via ubiquitination. However, when there's cell stress, for instance, DNA damage or presence of oncogenes, different types of metabolic or oxidative stress, P53 is then activated, and the activation is via phosphorylation sites that then releases it from its interaction with this protein here called MDM2 to facilitate processes such as DNA repair, apoptosis, cell cycle arrest, among many other these factors that we see here. So
excuse me, Victor. Excuse me. Please put the presentation in the presentation mode.
Okay, all right. Just a moment. Can you see it now?
Yes, that's much better.
Yes, that's okay. Thank you.
All right, thank you. Yes, so and the same can be said of APC and VHL. They are either mutated by inactivating mutations, or they are deleted completely from the chromosome, or they undergo an event called hyperphosphorylation that puts them in an inactive state, so they cannot respond to any of these stress signals. The implication of this is that damaged cells or oncogenes go unchecked, and once these accumulate over time, they are able to lead to tumor formation. So the next hallmark that I want to talk about is induction of angiogenesis. So what is angiogenesis? So angiogenesis simply refers to the formation of new blood vessels from existing capillaries. And this is important because it supplies the tumors with either oxygen, nutrients, and also removes metabolic waste. And we know that angiogenic switch is very important for tumors because they require these, they require nutrients and oxygens to grow, divide, and even spread to different organs. So at the onset of tumor growth, there's always a balance between factors that promote angiogenesis and factors that inhibit angiogenesis. But as the tumor continues growing, you can see the tumor cells start releasing more pro-angiogenic factors, and this might include factors such as VEGF, FGF, MMPs, and HGF, that then over time accumulate and lead to formation of more blood vessels that serve the tumor. However, it's important to realize that this process only occurs in a subset of tumors, because remember, as the blood vessels supply the tumor with oxygen, nutrients, as well as removal of metabolic waste, we can also exploit the same process to deliver drugs and therapy to target the tumor. So this process, I want you to understand that it doesn't take place in all tumors, but just a subset of tumors. And one of the main factors that the tumor cells release into the microenvironment that then promote angiogenesis, like I said, is VEGF, which in full refers to the vascular endothelial growth factor. And VEGF is the master regulator of angiogenic signaling in tumors. And it comes in many isoforms, VEGF A, B, C, and D. And upon binding to their receptors, that is VEGF-R1, VEGF-R2, or R3 on endothelial cells, they trigger intracellular signaling, which involves RAS, GDP, RAF kinase, MEK1/2, and ARK1/2 phosphorylation to promote endothelial cell proliferation, endothelial cell differentiation, and endothelial cell growth towards the tumor. So we have a couple of inhibitors that have been developed specifically against VEGF-A, or where we have bevacizumab here, it's a monoclonal antibody that only targets VEGF-A. We have another monoclonal antibody here that targets VEGF-R2, as well as a broad spectrum of what we call tyrosine kinase inhibitors that then target different receptors on endothelial cells that might activate the process of angiogenesis. So this form of treatment is very effective for renal carcinoma, that's cancers of the kidney, because they have been shown to express most of these receptors within their microenvironment that allows this treatment to be very, very effective. So the next hallmark that I want to talk about is activation of invasion and metastasis. So tumor metastasis simply refers to spread of the tumor from the primary organ to a distant organ, and it contributes to 90%. It contributes to 90% of cancer-related deaths, and this is basically the holy grail of cancer. If there was no tumor metastasis, then you would definitely be able to treat most, if not all, of the cancers that we have. And tumor metastasis is very specific and depends on the primary site of the tumor, because, for instance, you see melanoma tumors prefer to go to the lungs, to the brain, or to the liver, and a tumor like prostate cancer, for instance, might go to the bones and not any other place. Tumors like renal cancers might go to the lungs, to the liver, to the bones, and to the brain. But it's not always the case, yeah? So you might have a group of renal cancer patients where some of them only have lung cancer, others have both lungs and liver cancer, others don't have lung cancer, liver cancer, bone cancer, but only have brain cancer. And this means that the drivers of this process of metastasis is very different in that context. So how does the process of metastasis take place? So at the primary site, most tumors actually have an epithelial phenotype. This implies that the cells are really compacted together. They cannot move. Yeah, they have tight junctions that hold them together. However, once the tumor reaches a point where it's about to metastasize, these epithelial cells have to go a transformation called epithelial to mesenchymal transition. So the epithelial cells adopt a more mesenchymal phenotype, which is this kind of phenotype here. And this is associated with loss of these adherent junctions, changes in cellular morphology, so they become more spindle-like. That allows them to kind of intravasate into the bloodstream, go through circulation, and be able to leave the blood circulation to colonize a distant organ. And it's also associated with changes in gene expression, and mostly they express what we call proteases or matrix degrading enzymes, because whenever they are invading from the primary site, and also whenever they are colonizing a secondary site, they have to deal with a lot of matrices, so they need to break them down. And also another factor that changes is increased motility. They are able to move from place to place easily. So this whole process via which the primary tumor changes from an epithelial phenotype to a mesenchymal phenotype to colonize distant organ is what we call epithelial to mesenchymal transition, briefly referred to as EMT. So like I said, this process is the holy grail of cancer. If we could find a way to manage metastasis, then we would easily be able to treat many tumors. However, the process is not as simple as is detailed here. This is just a graphical representation of what happens. So the process from the timeline from the primary tumor breaking off to the colonization of a secondary organ might take ages, from say 2 years to actually up to 10, 15 years, depending on the primary tumor. So next is evasion of immune destruction. So we know that our immune system detects and tries to kill cancer cells because that's what they're supposed to do. However, most tumor cells have found a way to circumvent this process. So how do tumor cells recognize and evade the immune system? Number one, they don't produce tumor antigens. So we all know that for a T cell to recognize a damaged cell, an antigen must be presented. So tumor cells stop producing the antigens. Number two, they might produce the tumor antigen, but they lack the MHC molecule that might present the antigen to the T cell. And you see in these two cases, the T cell is active. Yeah, the immune system is active. The only problem is it cannot recognize the tumor cell. In the third case, you have both the tumor antigen present and you have the MHC molecule that is able to present the antigen. However, tumor cells release different factors into the microenvironment that then inactivate the T cells. So they shut down the activity of the immune system. So these immunosuppressive proteins that then block the activity of T cells include factors like interleukin-2, TGF-beta, program death ligand 1 and ligand 2. And you'll see why this is very, very, very important. So cancer cells are able to evade immune destruction by delivering signals that hold the immune cells in check. And we know immune cells, and mostly T cells, they express a protein on their surface called PD-1, which is a receptor, an immune checkpoint that guards against autoimmunity and inflammatory responses when it interacts with its ligands. So when we have an antigen, when we have an antigen present in our blood, the antigen-presenting cells like dendritic cells are able to present the molecule to a T cell receptor here, and this process activates intracellular signaling that then leads to increased proliferation of T cells, differentiation, increased T cell survival, and increased cytokine production. However, once they are able to deal with the problem, the same antigen-presenting cells come back and they present a molecule called PDL1 that binds to PD-1, And this one is now a regulatory signal. It's able to tell the T cell that, okay, whatever we were trying to clear, the damage is gone, so now you should stop working. So it keeps the immune system in check. So this leads to a second phase of intracellular signaling that leads to decreased proliferation and differentiation, decreased T cell survival, and decreased cytokine production. And so tumor cells have adopted the same mechanism. So they pretend to be antigen-presenting cells, express PD-L1, are able to bind to PD-1 on T cells, shutting down their growth, differentiation, cytokine production that might otherwise harm the tumor. And it's this process here that has been exploited for the development of new drugs, the ones that we now hear known as immunotherapy, which have been hugely effective in a subset of tumors such as melanoma, lung cancer, renal cancer, and colon cancer. However, this process is only effective in hot tumors. And by hot tumors, I refer to tumors with a high infiltration of T cells. Cold tumors such as pancreatic cancer, brain tumors, and ovarian cancers do not respond to these immunotherapies. So just to recapitulate what I'm trying to say, when you have a tumor cell, it produces an antigen that is recognized by the T cell receptor. But on the other hand, the tumor cell is also able to produce PD-1 that binds to produce PD-L1 that binds to its receptor PD-1, shutting down the immune system response, resulting in increased tumor growth. However, in the presence of anti-PD-1 treatment or anti-PD-L1 treatment, this interaction is inhibited. So it implies that the T cell is able to recognize the tumor, activating immune response and leading to decreased tumor growth. So having gone through some of these hallmarks, I thought it was important to just give you a brief example of how a tumor develops. So if we look at the case of pancreatic cancer, which is one of the deadliest cancers that we have globally, you have a normal cell, a normal pancreatic duct cell, and the first step that occurs is the mutation of the Keras gene. So Keras gene mutation in pancreatic cancer occurs in 99% of all the cases. So it's hugely addicted to this mutation here. And Keras mutation goes hand in hand with telomere shortening. And these two processes represent two different hallmarks. So one of them is sustained proliferative signaling, and telomere shortening corresponds to replicative immortality. Then the second step that occurs is the mutation of a cell cycle cyclin kinase inhibitor known as CDKN2, which then leads to evasion of growth suppression. And then in the third stage, you have the mutation of TP53 that occurs in 85% of the cases, which leads to evasion of growth suppression again. And then we have another gene that is again mutated, known as SMAD4, and it occurs in 55% of the cases. And this leads to sustained proliferative signaling and evasion of growth suppression. So between the first stage of Keras mutation to the last stage of, which is the SMAD4 mutation or deletion, this could take years. And this also explains why pancreatic cancer is very difficult to diagnose at an early stage, and also why it basically never, there are no biomarkers to help kind of manage the disease. So what's the take-home message here? I would like to tell you that cancer is not a homogeneous disease, even from the same subtype. For instance, if you look at breast cancer, you have different types of breast cancer. So if you look at it histopathologically, you can talk of lobular breast cancer, ductal breast cancer, inflammatory breast cancer, or a mixture of all these three. And these hallmarks of cancer are hypothesized. I should have replaced that with thought. They are thought to be necessarily acquired during the multi-step pathogenesis of these tumors. So they just don't occur at once, but it's a stepwise process that takes place. And then finally, different forms of cancer are dependent on different types of hallmarks, and this should be able to inform prognosis, progression, and response to therapy. So I come to the last part of my talk. So the research gaps in Kenya. So I did a PubMed search on using the search term Cancer Kenya, and I came up with 1,747 entries. And out of these entries, I wanted to find out how much of this correlates to breast cancer, cervical cancer, prostate, et al. And you can see from the figure on the left, cervical cancer and lymphoma are two of the most widely studied tumors in the country. But what was interesting to me is I wanted to also find out what kind of study type do we have or is happening in the country. And you can see that out of 1,747 entries, we have at least 600 studies that are based on epidemiology. Then you have a little bit of pathology. You have some that look at clinical trials. Then you have a few that focus on cytotoxicity, medicinal chemistry, and also on screening. And all these studies are very, very, very important. But what should really be worrying to us is that studies that focus on molecular profiling of tumors as well as signaling are very, very, very low. For instance, in the whole country, we only have 17 studies that focus on molecular profiling and 22 studies that focus on signaling. And why are these important? For us to be able to diagnose tumors properly in the country, for us to develop patient-specific therapies for Kenyans, we need to profile our own tumors. We need to understand the signaling mechanisms that happen to these tumors so that we can develop the right biomarkers for treatment, the right biomarkers for diagnosis, and the right biomarkers for signaling. So, and with this information, we can use this information and also compare it with information from Caucasian populations, where a lot of this has been done, to try and find areas that overlap and areas that are different. So the next thing that I wanted to talk about is just to give you a brief overview of the crude numbers of top 10 cancers in the country. And this is based from the Global Cancer Registry for 2018 and 2020. And you can see that in 2018 is in blue and 2020 is in red. You can see that in 2020, we have an increased number of breast cancers reported in the country. Cervical cancer is almost the same. We have increased numbers of prostate cancer. But here we observe decreased numbers of leukemia, decreased numbers of lymphoma, decreased numbers of stomach cancer, and also decreased numbers of liver cancer. However, I think we have to look at this data with a lot of caveats, because 2020, as we all know, we had a lot of movement disabled due to the pandemic. So the numbers might not be a true representation of what is actually happening in the country. And secondly, these numbers are based on the Nairobi Cancer Registry. But we have more than 47 counties in the country. We have more than just one county in the country. So we need to have a system where each of the counties is able to develop their own registries and then kind of boost these numbers up, because we all know that the numbers of cancers in Kenya are higher than just what I've presented here. And of interest that I also wanted you to have a look at is that even though lung, kidney, and pancreatic cancer are not part of the top 10 cancers in the country, you can see that from 2018 to 2020, we saw an increase of 18% of lung cancer cases in the country, an increase of 14% of kidney cancer cases in the country, and 10% increase of pancreatic cancer cases in the country. In terms of death, you can see that patients who succumbed to kidney cancer increased by 29% from 2018 to 2020, by 11% for pancreatic cancer from 2018 to 2020, and 11% again from lung cancer within the same time period. So this brings me to my next slide. Cancer research funding saves lives. As a Kenyan, I know that this is an area of funding that is really limited in the country. And for the first time last year, the government was able to put out a call specifically for cancer research. And if we look at this map of the United States, just looking at the 5-year cancer survival rates in the United States between 1977 and the year 2013. You can see, we all know that over time, the United States has increased its funding towards cancer. And you can see that this, for all cancers, which is this red mark here, in 1977, the 5-year survival rate for all cancers was about 50%. But you can see right now, it's increased to around 60 or 70%. And something like prostate cancer has moved from around 70% to around 98, 99% 5-year survival rate in the United States. All this is because of the funding that has been poured into the field to kind and understand what's happening with these diseases. So all kind of research is important. This is not only for molecular cancer research, it's also for diagnostic research, epidemiological research, screening research, clinical trials. So we need a wholesome approach towards this problem. A caveat that I have to say, a cancer like pancreatic cancer, you can see it's really not moved. It was about 2% in 1977, and now it's barely 10% in 2013. And even now in 2020, I don't think this figure has increased that much. So, as an organization, Richard at the introduction said that we work for, that I'm the chief scientist of Cancer Research Kenya, and our goal as an organization is to become a leading player in the precision medicine field, not only in Kenya, but in sub-Saharan Africa. And I'll tell you why that is important. You could have two patients, A and B, they both have stomach cancer, but patient A has got a mutation on this end, patient B has got a mutation on this end. If these two patients report to the clinic and you give them the same drug, they are not going to respond. One of them is not going to respond to that drug. So precision medicine simply means that we want to tailor-make drugs to target specific hallmarks present in the same tumor type, but in different patients. So it's not a one drug fit all, but it's different drugs to fit different types of patients. And this kind of research is largely missing in the country, and it's the gap that a cancer research organization, together with academic institutions such as Kenyatta University, wants to plug that hole. So what's our solution? We are proposing a different type of research model. So we want to see a situation where we have collaborative research groups that work on around major questions centered on a specific theme. I could give you an example before I explain what the figure means. Counties such as Kericho, Bomet, and Nandi County, we all know have got higher incidences of esophageal cancer. You go to Meru County, they have got higher incidences of stomach cancer. A lot of studies that have come out from these areas refer to they drink a lot of hot tea or they take mursik. And for the case of Meru County, they say they consume a high level of cut and mira and other things. Yes, that could be true. That could be true. That those lifestyles might actually be risk factors for these cancers. But beyond all that, there's an underlying genetic or molecular events that are actually happening in those populations that need to be answered. So you could have questions centered around such themes, so that you kind of really get the actual things that are happening in this group of patients, so that you can develop proper drugs for them. So what do we anticipate? We anticipate that as our patients go to the hospitals, either for clinical trials or for surgical removal of tumors, their clinical information is collected. Their cancers are characterized properly by pathologists from the day of onset, the kind of treatments they've got, any sort of mutations that they may have, and everything. And then we as researchers, because it's now a collaborative research, we as researchers will also take a portion of these tumors and we will grow these tumors in mice. This is what is called as patient-derived xenografts. And the advantage of this is that if you take if you take a biopsy from a patient and you inject the same in mice, it retains the same morphology. It retains the same. Genetic markers. It behaves as if it were in the patient. And why is this important? While you're treating the patient with a drug, you can also treat the mouse with the same drug. And if the patient is not responding, the expectation is the mouse is also not going to respond. But instead of subjecting the patient to another set of surgery and to remove the tumor in order to understand what is happening, you can simply sacrifice the mice, following all ethics approval and everything, every legal opinion set down, and actually analyze these tumors. And through this, you can do genetic screens, you can do high drug throughput screens to try and understand, is it a new mutation? Is it a new mutation? What's actually happening? Why is this patient not responding to this treatment? And so through that, you are able to then tailor-make new drugs that can then go into a second phase of preclinical trials just to find out how the drugs work and so on. And then these drugs can then go into actual clinical trials where the patient can receive the specific drug that was used to treat the tumor where we observed remission. So this is the kind of research that gap that is actually missing in the country. And for us to actually combat this problem, this is the model that we need to approach. Yes, epidemiological research is important. Yes, screening, clinical trials, cytotoxicity studies, medicinal chemistry, yes, they are important. But unless we do this kind of research, we are never going to get where we want to be. And I might sound insensitive right now, but we all know cases of Kenyans who have cancers and then they travel abroad, but most of them unfortunately succumb to this disease. Reason? The drugs that they go to get in Europe or in the US or in Australia, those drugs were never tested in our populations. So we don't know how we are going to respond to them. Yeah. So unless we start molecular fingerprinting of our own tumors, understand the disease pathways, understand the genetics around it, the epigenetics around it, and develop our own drugs, this problem is here to stay and it's not going anywhere. And it can only be done through collaborations between patient advocates, research institutions, academic institutions, the natural government, philanthropists, and so on. So with that, I would like to finish my presentation and acknowledge and thank Douglas Anahan and Robert Weinberg, the authors of the Hallmarks of Cancer. The institutions that I work for, the Integrated Cancer Research Foundation Kenya team, and that's our website, the Integrated Cancer Kenyatta University Research Office, and all of you for listening. Thank you.
Well, thank you so much, Victor. That was very invigorating talk. We have enjoyed it, and I think in our special way, we have already clapped our hands in the current online spirit. I think, colleagues, we have listened to Victor talk, and of course, breaking down the 5 hallmarks of cancer, ranging from the signaling, evasions, of course, angiogenesis, or we're talking about activation of the invasions, and of course, avoiding the immune system. These are very interesting, and probably, Victor, even as colleagues are putting their questions across, it would also be very nice to probably hear what could be other triggers or risk factors for some of these various mutations that happen at different points, and especially on the rate limiting steps, if it is a mutation. So my question would be in what are the leading currently the risk factors that are triggering a lot more of these mutations in these cancerous cells? And of course, also just picking your brain on what is the extent of using the RNA silencing mechanism and the prospects for the genome editing as a platform to look at new cues to getting new drugs. But those are just the two that I'm throwing your direction. Colleagues, you can unmute if you've got a question, and then we pick a little bit more questions, and he can answer them in mass.
Okay, so maybe if I respond to Richard's first question, what are the triggering mechanisms of this mutation? So, for instance, in breast cancer, for instance, you have these DNA repair genes such as BRCA1 and BRCA2, which are supposed to be like important in checking for transcription correctness, replication correctness. If these are mutated, it means that those problems that may happen when the gene is replicating or when you're having transcription, they are not under check. But one mutation is not enough to drive one mutation, or one single event is not enough to drive the formation of cancer. So what happens is over time, as these mutations accumulate, then you are able to have like a cell that has gone wrong, over dividing, and then actually capturing acquiring a whole organ for itself. In other cases, for instance, in pancreatic cancer, we don't know what is Keras mutation. Is it spontaneous? Nobody has really studied about that. So for some cases, you can link the mutation to the absence of a DNA repair gene, or to defects in in things like RNA polymerase. They're not able to do the process of transcription properly. But for some instances, you can't place your hand on why these things happen. I hope that answers the question.
Yes, it does, but of course on the prospects of RNA silencing and probably genome editing to try and look at this, what's your thought?
Yes, genome editing is a very interesting technology that is actually, we know that Jennifer Doudna and her colleague won the Nobel Prize for it last year, especially CRISPR-Cas. However, it's not as direct. It's not as direct as we think, because we have redundancy in the genomics in our genome sequences. So you may target a particular gene with a particular sequence, but what if that same sequence is important or is found in another gene, or is important for another marker in the body, an important protein in the body. So that's the caveat that we have right now, that you don't know what you're targeting. And I could give you a very good example. When we use CRISPR-Cas to kind of silence genes in cells that we use for our research, we then make single cell colonies from each of these targets. And for the same genetic target, you see the cells behaving differently. And when you do proper follow-up, you find that there was a second hit somewhere that you are not aware about. Yeah? That there was a second hit somewhere that you are not aware about. So this is the danger. This is the danger of gene silencing. Of course, there's a lot of research going on, and maybe in the near future, we might have effective RNA silencing therapies. Yeah, we've already seen one. We already see one with this, with the development of this RNA vaccine for COVID-19. So maybe, yeah, who knows? Something is in the offing soon.
Yes, thank you, Victor. Colleagues, any question for Victor? Victor's questions come your direction. Also, I would want to pick your brain on regenerative medicine. What do you think around that? If we advance it to the level of regenerating very critical organs, and we think that shouldn't we think more about that to replace this particular localized cancers, especially those that have not undergone metastasis, which makes it very difficult for such kind of attempts because you have to replace every organ anyway. But for the localized sort of tumors, wouldn't you think that also advancing heavily on the regenerative medicine and with a view to having tissues sort of mounted back would be a good approach? And if so, the diversity that comes with different genetic variations, especially in different places, Africans and other parts of the world, would that be a challenge from where you sit?
I mean, we are already doing it for bone marrow transplants. But regenerative medicine also has got its own ethical issues. Yeah, because where are you going to grow the organs? Is it in say pigs or in apes or yeah? So these are things that these are things that can happen. But some of the caveats that we have is the ethical issues around it. So are we going to be growing, say, for instance, pigs for food or pigs for regenerative medicine? For instance, that's just an example. Yes, but I think it's an area of that is also actively being researched, trying to find ways of overcoming these challenges. And one of the things that is actually now happening is 3D printing of organs. Yeah, you have 3D printing of organs. How that works, whether it will work successfully, is still a matter of debate. But yeah, there's tons of research happening out there on all those questions that you're asking.
Thanks, Victor. I'm now reading from some chat from Festo is asking whether we have capacity as a country to undertake molecular fingerprintings of tumors. Yes, we have the capacity. Yes, we have the capacity, and I will tell you why. If we only rewire our thinking, if we only start to understand the importance of this kind of research, yes, we can do it. Yeah, and I'm going to sound a little bit political here. If we can spend 65 billion on imaginary dams, 65 billion is enough to build 3 or 4 cancer research centers in the country and fund them properly and attract talent from all over the world, Kenyans and internationals, to come back and actually do these things. So yes, we can do it. There's nothing that stops us. I mean, we are doing those kind of things abroad here. What is the difference? They're simply funding it. So since independence, it's only last year that the Kenyan government was able to put out its first cancer-specific call. It was only last year that they put the first call. So yes, if we put more pressure on the government, if we put more pressure on our legislators, yes, these things can happen. Who knew that Isipe would grow to be what it is? Ilri would grow to what it is. Kemri to grow to what it is. It's because of just pressure. So I believe everything is possible. Yes, thank you. There is a general, really, question from Joyce asking very blunt question. Is there really a cure for cancer? That's a very interesting question, of course.
Yes and no. So for localized cancers, that have not spread to a different organ. You simply remove them by surgery and you're cured. And you can live a better life, longer life. So like I said in the presentation, the holy grail of cancer is metastasis, that is spread into a different organ. Once that process has started, then now we are in murky waters. Yeah. So surgery has worked for a long time and it's still working, and it's still the gold standard when you walk into a clinic and you say I have breast cancer, or I think I have liver cancer, or blah blah. The first thing that they do is, can we surgically remove it? So if they can surgically remove it with proper margins, yeah, you are good to go. But for metastatic cancer, that's where you have the problem.
There is another question from Gideon. Gideon is saying that he's been to several referral laboratories where cancer markers tests are done, but with all those, Kenya seems to not to have the experts and equipment to detect, diagnose, or even treat cancer. What is hindering us, and especially the government, from having such experts and, of course, machines?
So one of the problems that we have, not one of the problems, but one of the limitations that we have in the country is that for a long time we were exposed to infectious diseases. So all our money, all our capacity building and everything went towards infectious diseases. Things like HIV, TB, and also say malaria, though it's not infectious, pneumonia, those kind of diseases. So if you look at that area of research, we have a lot of experts. We have good equipments. We have personnel that can handle that. For cancer, we are a little bit late. We are a little bit late because I can count on my fingers the number of Kenyan cancer PhD holders on cancer biology. We are very few. And this is also one of the reasons why we started this Cancer Research Kenya, because we want to come back and see whether cancer training, cancer biology curriculum can be added to some of our biomedical courses to introduce students early enough to this disease, and even have specializations, for instance, at master's level or even at doctoral level, where people can actually do a program on cancer biology and specialize in this kind of thing. So yes, we are a little bit late, but we will get there. We'll get there. All we need is just to be united and make sure that we press the right buttons with the government, and we will see how that goes.
Yes, Victor. A question from Eunice says, due to the differential, probably community perception of cancer as a disease, to what extent has that affected probably the cancer research, and what can be done? And also another question is, what is the best next step, and where can academicians and clinicians really and researchers start battling with this cancer research and probably other aspects of the same?
So where do we start? For instance, what we are doing right now, yeah? What we are doing right now, talking about the disease, presenting it from a different perspective. Because yes, a lot has been happening in cancer in Kenya, but mostly it's on the side of treatment, screening, patient advocacy, and all those other things. And that's important, yeah? That's important. But for us to actually have real answers, we need to go deep into molecular research. Yeah? Molecular research will help us understand what is really happening with tumors in the country, and even in the region at large. We would see whether communities that are related genetically tend to suffer from the same tumor types or not, and so on. But what is limiting us is that, as usual, in Kenya, you have non-experts speaking about a topic that they have no idea about. Yeah, and that's the narrative we need to change. We need to have people, like-minded people, spearheading this kind of initiative. It's not a one-man show. It's not a one-woman show. It's a collective initiative, and it's also not about competition, because you realize that, again, because of also the publish or perish nature in our academia, people are competing to do research instead of collaborating to do research. So if we can have, and that's why I even stated in one of my final slides that we need collaborative research groups that try to answer a bigger question but different segments, so that when they come together, you have the whole picture of the idea. And then thirdly, in the absence of real research, that is why you have things coming up like hot tea can cause cancer, drinking mursik can cause cancer. I'm not saying they can't, maybe they do, but I don't think it's very definitive. Yeah? So in the whole process, we also need to be training a lot of people on scientific journalism, so that you just don't report things like that. They must undergo rigorous peer review before they put on a newspaper, the Nation, the Standard, the Star. Because people trust what they read most of the time. They're like, oh, I saw it in the Star, I saw it in the Nation newspaper. And in most cases, it might not be. So those are the areas that I think we could deal with, so that we can collectively combat this problem.
Yeah, thank you, Victor. And I think just rephrasing Eunice Nyamasio's question is that probably most communities have their concerns, especially with breast-related cancers, because of how, especially, we were brought up. It takes a while before somebody can expose their breasts and actually report that something is really happening here. And to what extent has that really affected the whole process of diagnosis for this disease? That's a concern. Even as you ponder about it, I would ask if there are more questions. If none, then we will probably give you just a minute or so to just give us your final thought, and then I can close this. Colleagues, if you allow me.
So, response, is somebody asking a question?
Well, not now. I think we're just giving you the opportunity to throw in your parting shots, and then I can do the closing bit.
Yeah, so I would like to thank everyone who attended the meeting today, or the seminar today. I'm sorry if it was a bit technical, but that's my field of expertise. That's what I do, molecular cancer research. So I wanted to introduce you to the field. So next time, if we are having a specific topic on probably this gene is important for this particular cancer, if I speak about metastasis, you'll probably have an understanding of what I'm talking about. If I speak about things like P53 or VHL protein, you probably have an understanding of what's happening. That being said, I would like to also say that I would not be here today without the involvement of our organization, that is the Integrated Cancer Research Foundation. So if you go on Google and just type Cancer Research Kenya, you'll find us there. We are a young organization. We are trying to bring real molecular work in the country, and with your support, either through volunteerism or donations or anything that you can do, yeah, we are glad to receive that help. And third, we are also trying to see whether the government can increase cancer-specific research funding. So this is something that you can also champion for. Yeah, this is something that can also help champions. The more we increase funding, the more we are able to understand what is happening in our communities and develop local solutions. Yeah. So other than that, I would just like to thank everyone for attending the seminar, and yeah, we will.
Yeah, thank you. All right. Thank you so much, Victor. Of course, Jackie is throwing in a question, wondering whether there are food components that can trigger mutation. But of course, that's just for a very interesting question, though. But colleagues, even as Victor thinks through that, and probably we're summing this up, we are in deep discussions right now with the organization, Cancer Research Kenya, in a view to having more specialized seminars like this. We are already planning for almost 6 or so series seminars from that organization from Oxford, and this is coming from our fellow, Kenyans who are abroad. As a directorate, we are now keen into looking into what are the specialized areas that would trigger, and I would really want you to be involved in this, in getting to know what are the critical areas that you would want us to source for experts to give a seminar or two on the same. This is very critical so that we keep abreast with the emerging trends in research. And of course, this can help us start identifying the areas that are critical for us to grow as researchers, and also just to network with the rest of the world. Half the time, again, colleagues, this demand for now multidisciplinary and also multi-institutional and now multi-regional and all this. So I take this as also an opportunity to link up. Victor's email is available. Just in case you need some networks around that institution in Yale University. That would be great. So as a directorate, we thank you for availing time and, of course, being very active on this. I know Victor had warned me that it's going to be a bit technical, but we like it as well, that it triggers our interest on the same. I know you're now running from the hardcore research to probably also advocacy, which is research-based or evidence-based, and that is an area that would be very, very interesting also for us. So we've got a series of other seminars, colleagues, that will be throwing your direction going forward. So once again, as a directorate, we are very grateful and we are here to serve you. And if there is any area that you feel that you need to get experts, that's an area that personally I'm very critical in. And of course, sourcing that from our own diasporians who are out there. And because there's a network, we're already linking with the Ministry of Foreign Affairs to give us a database of all of them. If out there, at least for Victor, at least they already have an organization that is trying to bring all of them together. We are looking forward to having others in different departments, in different areas as well, be it even in business or even in climate change. All these emerging trends, areas are very critical for us. So without sounding redundant, allow me to say thank you so much. I thank the team here at the Directorate. My name is Richard Oduor. I serve currently as the Director of Research Support and Dissemination, and I'm an Associate Professor in Molecular Biology. And so it was very interesting to hear my former student that time, Oria, talking very nicely, and congratulations once again, Oria, and we are looking forward to having you back as soon as possible. Thank you, colleagues. Allow me to sign off. I know questions are coming on, but you can pick this up directly now with Victor. He's available. And until next time, thank you so much and God bless.
Okay, so I can have another 15 minutes. If anybody has a question, I can respond to.
Yeah, that can now be just an informal interaction now, yes. Colleagues, if you still, but you are free to leave now. I don't think so. Probably just if you want to ruminate with the food component bit, whether there's any food component that can also trigger mutation. That was a question. Then there is also a question from Nora. Can a doctor determine if a swelling is cancerous or not just by looking? Yeah, interesting, Madeka. Does one have to do biopsy to determine if a swelling is cancerous? That's also another question.
Yeah, I think a biopsy is a definitive way of determining whether a swelling is cancerous or not. Yes, you can, unless you have magical powers, yeah. But I think you have to do a biopsy. Important. So, do food components have things that trigger mutations? It depends on where the food is grown. If you're eating food rich in heavy metals such as mercury, lead, arsenic, then yes, it's so there's always something behind. Say, for instance, eating this type of food. It depends on what's in the food. Yeah.
All right. So I think colleagues will bring this to probably a total close. Just in case you didn't capture Victor's email address, you can reach out to us. We can always share it directly to you, but also to appreciate your time. Thank you, Victor.
Okay. All right. Bye, everyone.
Bye-bye.